Our speaker is Dr. David Kaplan. Dr. Kaplan is professor in the Department of Environmental Engineering Sciences within the Engineering School of Sustainable Infrastructure and Environment at the University of Florida. It's a little bit of a mouthful. He's not only a professor, he's the director of the H.T. Odum Center for Wetlands and the associate director in the Center for Coastal Solutions. He has a BS from Cornell University and a PhD from the University of Florida. Research in Dr. Kaplan's lab focuses on linkages among the hydrological cycle, ecosystem processes, and human activities across a diversity of global ecosystems with a goal of developing new scientific knowledge and engineering tools to advance natural resources, conservation, and management. And today he's going to talk to us about mangroves increasing in abundance in the Gulf of... Thanks, David.
Thanks, Laura. Thanks for the introduction, and really appreciate the invitation to speak in this series. I'm going to share my screen. We will try this and make sure it works. Continue to share. We looking good?
Looks great.
Excellent.
Thanks.
Okay, thanks so much again. I want to acknowledge first and foremost, I need to acknowledge the person who did the lion's share of this work, who is a recent PhD graduate from my lab, Yi-Yang Calvin Kong. He graduated in August and has gone on to bright future and continues to be excited about this work. I'll share some of the latest outcomes of Calvin's work right at the end, but this is some work that we've been developing for a couple of years now with colleagues at the USGS and also with funding from the USGS and IFAS, SNRE, and the Nature Coast Biological Station. So basically, we're talking about mangroves moving and changing sort of where on the landscape they exist and kind of what they look like in those locations. So I want to start with a little bit of a story or a picture, pictures being worth 1,000 words. And if we were all together, I would say, hey, does anyone know what we're looking at in this picture? And I won't encourage everyone to unmute because you just got good instructions to not do so. But we're actually looking at here is off of Cedar Key, and this is January 2023, and we see all three species of mangroves that exist in Florida, the black mangrove, red mangrove, and white mangrove. And throughout this talk, I'll try to refer to them by black, red, and white. Sometimes the Latin name will come in there. And so here we have this picture from January 2023, and I'm not going to tell you exactly why that date is important yet, but I'll pop up another picture, and this is a picture from Apalachicola Bay, further north out in the Panhandle. And I've got two pictures actually of the very same view, July 2022 on the top, and then January 2023 on the bottom. And here, the pictures are both showing red mangroves, but they're showing a difference in sort of how this species responded to something that happened between July 22 and January 23. And I'll give you sort of the spoiler alert here, which is that there was a really severe freeze event over Christmas 2022. I don't know if you remember it, but the Christmas freeze of 2022. And it really piqued our interest in this larger question of how mangroves are changing their abundance and their composition and community composition along the coast. So really the story that I'm going to tell today is a tale of three species and two strata. And by species, I'm talking about the black, red, and white mangroves. And by strata, I mean basically tall and short. So tall and short mangroves. And, you know, I'm going to talk more about this later here, but you might see that this red mangrove in Apalachicola, the tall stuff all died and the short ones survived. And so I'm really going to try to draw some species and form specific understanding of how mangroves are changing on the coast. So if you had to, like me, you had to read back in high school, A Tale of Two Cities, and with my sincere apologies to Charles Dickens, that one starts with, it was the best of times, it was the worst of times. And if this novel was rewritten by Florida mangroves, they might say it's mostly the best of times. This is sort of the take home here as we experience climate warming and see mangroves expanding in their extent and their range, abundance, and biomass and all the rest. So that's basically the take home. If you don't remember anything else, it's that mangroves in Florida are feeling like it's mostly the best of times, notwithstanding things like, you know, potentially being cut back for development and other things. But as far as climate goes, looking pretty good. Okay, so zooming out now, just to think generally about how climate change affects things that we see on the landscape, the organisms that we see. We're all familiar with graphs like carbon emission scenarios and projected global temperatures under different renewable energy scenarios. And with warming temperatures, we expect to see species range shifts. So this figure on the right shows that there's a variety of ways that species might adapt to warming temperatures, everything from going extinct on the bottom left of that figure to expanding in range. They can sort of move upslope, they can move up latitude, and they can basically rearrange based on habitat suitability. And I think the important thing to think about when we imagine what is a species doing is that climate warming is affecting individuals, like uh, specific individuals. It's affecting species, affecting whole communities, and moving up to ecosystems and even landscapes. So the picture on the right there is the Carolina warbler, the Carolina chickadee, basically habitat suitability going much further, much higher habitat suitability into the Northeast over time. And species all across the globe are experiencing this change in range. And it could be moving north, it could be moving south, it could be moving upslope, downslope, wetter, drier. And so an interesting question is what happens when something foundational like mangroves begins to move and how that affects many other related ecosystem services, many other related species and habitats. So we have this idea of tropicalization. It's defined as basically temperate ecosystems that are being replaced with poleward moving tropical organisms as temperatures warm. As it gets warmer, it's not just the mean temperature that goes up, but we get a reduction in the frequency and the intensity of extreme winter events. So fewer freezes, fewer really deep cold snaps, and that allows the poleward expansion of cold sensitive species. So we're in the northern hemisphere, so poleward is basically moving north. In the southern hemisphere, poleward, of course, would be moving south. But the question we want to understand as ecologists or as natural resource conservation, as managers, is how are these species going to change over time? Is there going to be sort of no response? The species going to stay where it is, like in that thing labeled D, that line on the bottom? Are we going to get big steps and jumps in a species like the one labeled a smooth transition, or perhaps like a sawtooth, where things will move north and shift south and move north and shift south? And a lot of authors have looked at this in specifically in terms of what mangroves are going to do. We've seen mangroves expanding poleward across the globe, and their extent and their range expansion is affecting everything from habitat, of course, like a tremendous habitat for birds, for fishes, and vertebrates. Coastal protection, one of the greatest ecosystem services mangroves have to offer, of course, come along with that mangrove expansion. So we see the changing landscape of the coastline. We see blue carbon, potential increases or changes in the storage of carbon at the coast, and the list kind of goes on and on. So the question is, if we really want to understand what's going to happen in the future, what our ecosystem state's going to look like, how are we going to manage coastal ecosystems in the future, then we need to understand both the areal change, like the extent, the range shift, as well as what is happening to specific species and specific strata as winter temperatures are changing with climate. So that's sort of our challenge here. Well, it was Calvin's challenge, really. It was his dissertation. And so we're trying to connect the dots. We know that climate change is occurring, that there's this warming winter temperature regime. We want to understand how that affects mangrove vegetation. So we want to understand how do mangroves vary in space? What are the species compositions? How much biomass on the landscape? How are they growing? And critically, it's not just mangroves writ large, but individuals, population, and community scale where we see that spatial variation. We also want to understand how mangroves are changing over time. Are they expanding? Are they resisting? Are they basically resilient to climate shocks? And is that happening sort of at a freeze or at scales much longer, like decadal warming? And again, across scales. And then Calvin also looked at this effects on mangrove soil, but there could think of any other knock-on effect from habitat to coastal protection and on down the line. So the way that we organized Calvin's dissertation was around three basic chapters. I'm going to talk about just two of them today, where we look at the temperature sensitivity of mangroves across that individual population and community gradient, and also how mangroves responded to that freeze event in Christmas of 2022. I might have a word or two about the soil carbon at the end. So starting kind of like a zoom into the Gulf Coast, we've got this mangrove range expansion hotspot. If you work or live on the Florida Gulf Coast, you've probably observed it in recent years and decades, but it's a very dynamic zone. It's sort of this interface between the tropics and the temperate zone. We've seen this rapid poleward expansion. And what's really neat about Florida being such a, it's a very long state, a lot of latitude, is we have a really strong temperature gradient from, from hot to very hot. But we're able to build something that we refer to as a chronosequence. And I want to describe this a little bit more detail here. But if you are in Cedar Key, or if you drive out to Cedar Key and went off the coast, and you might find a mangrove stand that looks like this, really tall, relatively dense black mangrove stand, 8+ meters, 25 some feet high, really strong, robust mangrove ecosystem. But if you got in your time machine and went back 5 years or 20 years or 30 years, you would see a different story. And you would see the development over time of a marsh dominated system into one where mangroves were encroaching, and to the point now where they are quite dominant in much of the area around Cedar Key. If you went to the north to Apalachicola Bay, you'd see a different story, dominated by marsh cover, Spartina and Disticlus and Juncus, with a couple of red and black mangroves popping up. And what's interesting, of course, is if you went down further south, you would see a different story of mature mangroves that have been developed not for just 20 or 30 years, but more like 50, 60, 80, or maybe even several hundred years. So this idea that looking over space across that latitudinal temperature gradient kind of gives us an idea of what's happening potentially in one space over time. And we call that a chronosequence. And the question is, well, here we are today in Apalachicola Bay. It's looking like sparse dotted mangrove coverage, but in how many years will it look like what's going on in Cedar Key? And so that begs the question, like in the bottom left, what is the pattern, the process, the rates, and the drivers of mangrove range expansion along the Gulf Coast? So this is the result of that first chapter. We looked at the spatial variation across the entire Gulf Coast, and there's a picture of Calvin in the field with Hannah Sackles, who is his primary field assistant extraordinaire, and we were looking at spatial variation in mangrove structure across the coast. In other words, looking sort of back in time as we went further south. So the research questions, there were three of them. How do mangrove community composition and biomass vary with the winter minimum air temperature? So this is the thing that knocks mangroves back, is that potential low temperature freeze event, both its magnitude and its duration. Are there, or what are the species-specific responses of populations and individuals across that temperature gradient? So do white, black, and red mangroves respond differently? And with that understanding, can we predict how mangrove ecosystem structure will change under future warming scenarios that we know are coming? We're living through them. So Calvin always used to give this slide and said, if you got in a pickup truck right now, you could go visit all of these sites. There's 18 of them across the Gulf Coast of Florida that we visited, and they're shown in stars and circles here. I don't need to get into all those details, but they're mapped on this red-blue color gradient. And the red-blue color gradient is the minimum air temperature recorded at each of those pixels in the last 40 years. You know, this very strong gradient from warm to cold in the south to the north. And if you got out of your pickup truck and you kind of looked at the different sites out in Apalachicola Bay, you'd see dominated by salt marsh with a few spotty red and black mangroves. If you went out to Wakasasa Bay, you would see a really tall stand of black mangroves. And as you went further south, you'd have both red, black, and some white mangroves, but you'd see real dominance by really mature, large, thick, very dense red mangrove stands. And so this is the set of sites that we went out to and set up transects to measure the structure of individuals, community, and population. At every site, there was a 100-meter long transect, and each one had basically a 100 square meter quadrat. And in each of these plots, these circular plots, we measured tall trees, short trees, and they were defined by the literature. We took soil samples, we took pore water samples, and we looked at percent cover, we looked at abundance, we looked at size, crown spread, crown dimensions. Within the plot, this was a sort of statistical analysis that allowed us to do things like percent cover. But as Calvin was out there, he also noticed that you didn't always get like the champion tree when you went out and set up this transect. We set them up randomly. And so we also added to this what we called a synoptic survey. And this was for a fixed time and a fixed radius. Calvin and Hannah would go out and visually look for the 5 tallest trees of each species. So not only characterizing the abundance, the above ground biomass, the percent cover, but also looking at basically what is the growth potential for the tallest trees in each of these locations. So we had the plot based and we had the synoptical study. So I'm just going to jump right into the results for this first question of how mangrove community changes with this air temperature minimum. We're looking at mangrove coverage and canopy height here. So on the left is mangrove cover, on the right is canopy height. And what we see is they show a threshold response to minimum air temperature. I'm going to attempt to get the pointer. Let's see if there we go. And so what do I mean by threshold response? So for a threshold response, I mean something is changing after a certain point. And in each of these graphs, we have a solid black line that is aligned along a certain temperature. Temperature minimum is on the x-axis, mangrove cover on the y here. And there's a place of inflection where basically you can split the population up into something that is high mangrove cover, and then it begins to change. The dashed lines are places we call the area of maximum rate of change. It's kind of like the zone of where that change occurs. So a couple things that we can see here. We can get the specific temperature thresholds at which we see mangrove cover decline, -9 degrees for mangrove cover, and where the canopy height begins to decline, which is -8.4 degrees Celsius for canopy height. The other thing we can get from this is we can look at the zone. The threshold zone is wider for coverage than it is for canopy height, basically meaning that the height of a mangrove canopy is more sensitive to that temperature minimum than is the coverage. In other words, as you're driving from south to north, you'll see a more stark change in canopy than you will see in cover. And so we can begin to think about as temperatures warm, how are both mangrove cover and mangrove structure, in other words, height, going to change over time. We can also look at the overall biomass. So how much actual woody biomass in the above ground in the mangroves themselves are being stored on the landscape. And we found that tall trees contribute to above ground biomass in this sort of linear fashion with Tmin. So again, the minimum temperature is on the x-axis. On this, the black dots are the tall mangrove above ground biomass. Basically, how massive is each individual tree? And on the second y-axis in green is basically how many are there? How many individuals are there? And if you kind of multiply them, you get the total biomass. But for the tall trees, we saw this very strong and significant linear increase that as you get warmer, in other words, as you're driving south, latitude is declining, you have this very strong response of above ground biomass and abundance. We don't see the same thing for the short mangroves, basically no significant relationship between Tmin and either above ground biomass or total abundance. And when we add them together to get the overall mangrove community above ground biomass, we see it's driven really by tall trees. And we have this very strong linear relationship. As you go warmer and warmer, you get a larger above ground biomass. So we see in the previous slide, we saw cover and we saw tree height. In this slide, we see the overall above ground biomass being strongly dictated or driven by minimum temperature. So that's sort of at the scale of the whole community. Now, what about these populations? How about at the different species specific responses? So here we're able to look at red, white, and black mangroves separately. And they're all plotted here, but I've got two plots. One is tall mangrove abundance on the left, and then it's tall mangrove percent cover on the right. And there's a lot of dots there, but I think one thing I want to make sure I say is that the black dots are for black, the red dots are for red, and the sort of grayish dots are for white mangrove because white dots don't show up. But we see is that at these middle temperatures, these middle latitudes, we have black mangroves basically being dominant. This is usually the species that colonizes first. They are pretty robust to freeze events, and we'll see that in a moment in the after post-freeze assessment. We see them being sort of dominant in the middle latitudes, but further south, red mangroves begin to dominate. So basically, red and white mangroves increase linearly with that Tmin, but these black mangroves have this sort of hump-shaped behavior, and it begins to help us understand, will those mangroves that pioneer, those black mangroves that pioneer as the temperature warms, will they remain in place or will they become less dominant as red mangroves move further up the coast? We also looked at this. This is again for these synoptic tallest trees. So these are not within the plot, but these are the tallest trees across the entire site. Very strong mangrove height and above ground biomass response to T minimum. So tree height as a function of T minimum, strongly linear for all three species, and the same for above ground biomass. For tree height, basically all three of the species had the same slope, meaning for each increase in temperature, you would expect the same increase in tree height. For above ground biomass, we saw it was a bit steeper for black mangrove than for red and white. In other words, for every degree increase in T minimum, you'd have more of an increase in black mangrove above ground biomass than you would for the red and the white. So it's a snapshot of the results, but basically showing that you can look at the whole community as we did previously. We can separate it out by red, white, and black mangroves. So look at the populations. And we can try to understand how these relationships, again, all scaled across this winter temperature regime, might help us predict what mangroves do in the future. So this is our third question. How will mangrove ecosystem structure change under future warming scenarios? Basically, all that information on the left was built from field-based relationships. Field data, measuring trees, measuring how big they were, how many there were. And because they were ranging over such a large temperature gradient, we could use them to understand how cover and canopy height and above ground biomass might change in the future. And so we did that for three scenarios: the current baseline temperature, a moderate warming scenario of 2 degrees centigrade, or an extreme warming scenario of 4 degrees centigrade. And that's what this looked like. The first thing we see is that circled in green, there's the dramatic increase in mangrove coverage and height, especially at its northern range limit. So each of these little pixels is basically the maximum mangrove percentage within that pixel currently on the top row, and then under moderate and extreme warming. And we see that basically coverage is expected to increase a lot over time. I'll give the numbers in a moment. Canopy height is also expected to increase. The tall trees in around Cedar Key being 20 to 30 feet tall versus the little shrubby ones in Apalachicola. If the relationships hold, then we expect that be a very large scale conversion in the structure of mangrove composition and canopy in the northern range limit. We also see that red mangrove. So right now, black mangrove is, co-dominant across the Gulf Coast, but it's more dominant in the north than is the red. As temperatures continue to warm, we expect that red mangrove will come to dominate a larger proportion of Gulf coastal watersheds. And so that will become the dominant further and further up the coast. And here are those numbers: a 16% cover increase overall, a decline in black mangrove, increase in red mangrove, increase in canopy height, and a very large increase in above ground biomass. So this is what we expect under extreme warming. And extreme and moderate, you'd sort of take your pick. 4 degrees centigrade warming is an extreme prediction based on the range of RCPs that are currently being worked on. But if we get there, this is what we expect. So a couple of conclusions about this portion. Basically, this idea of looking at spatial variation in mangrove structure across the Florida Gulf Coast and thinking of it as a chronosequence, basically looking at location as sort of a proxy for time. First, we found that mangrove communities had this threshold type response for cover and canopy height, but they had a linear response for above ground biomass. Again, these field-based measurements allowing us to predict future mangrove cover structure under different T minimum or different climate scenarios. We found that red mangrove may take over the dominance from pioneering black mangroves at the higher latitude. So black mangroves may show up first, and then red mangroves may come and take over dominance. And we found, again, increase of mangrove cover, height, especially at its northern range limits, and again, this increasing dominance by reds. We were surprised at first to go to Apalachicola and see that there was a real mix of both red and black mangroves. And so we're already seeing some colonization by red mangrove all the way as far north as Apalachicola Bay. All right, so that was the first part of Calvin's dissertation. He was really excited to get those results. And then we were kind of, I guess it's not great for mangroves, but I think we were perhaps blessed with this Christmas freeze, because all the data that I just showed you was all taken in the field season before, basically in the field season of 2022. We had this December Christmas freeze, and it was actually an opportunity to go out across these sites and look at what happens when you get a freeze event at sites that you just studied species and strata-specific responses. So this was moving to the next chapter in Calvin's dissertation. It was published last year in the Journal of Ecology back in October. And we were able to look at mangrove freeze resistance, basically how good were different mangrove species at resisting cold damage, and then how quickly they came back. So looking at their resilience. Zooming out a little bit, we understand that a lot of species have some sort of freeze mediation. If you're a gardener, you certainly know this, that you can only plant certain things in certain zones. We've learned, as the community has learned over time, that mangrove distribution and structure is controlled not by average winter temperature, but by the freeze regime. And as climate warms, that freeze stress is alleviated, and this is what has allowed mangrove range expansion. There's been a fair bit of work on this on black mangroves in Louisiana, less so in Florida, and certainly less so across different species and across different strata. So our idea is we want to understand mangrove freeze resistance and resilience, not just writ large, but what are specific species doing and how are they doing it? This is the picture I showed you at the beginning. It's just sort of rehashing that here. You know what? There was our black mangrove. It did really great after that freeze. The red mangrove took a little damage, but was pretty much okay. And the white mangrove really bit the dust. And it was amazing for Calvin to be able to get this picture all in one place, seeing the three species all responding differently, same location, same event, and obviously a species specific difference there. On the right, this is red mangrove up in Apalachicola and healthy in July. And then after the freeze, we see that the entire an individual of this tall mangrove individual died, but there were small seedlings at the bottom, these short ones that survived. So we wanted to quantify this. So I'm going to show a few pictures just to sort of show what these species look like after recovery. So I've got black in the top left, white mangrove in the bottom left, and then red mangrove on the right side. And we found that all three species were able to resprout and even flower and fruit after 1 year of recovery. And this was very well known for black and white mangroves, which had been known to resprout. But for red mangroves, it's a very rare, and we caught actually only out of hundreds of trees that we tagged and looked at and went back to see if they resprouted. Only one red mangrove resprouted. It's very rare, very rarely seen in the literature. So, okay. And we found flowers and basically reproductive success for all the species after the freeze event. All right, so what were the questions? We wanted to understand the species specific low temperature thresholds. In other words, how cold does it have to get for each of these species to have leaf damage or mortality? And then how quickly can they come back? We wanted to understand how that varied both as a function of temperature, as a function of species, and as a function of short versus tall trees. And we wanted to use this to understand what is the future warming trend, tell us about the freeze regime and how it's going to affect that mangrove poleward expansion. To get at this, we looked at the same set of sites. The map that I showed you before, I'm going to change it a little bit. This is the minimum temperature during that Christmas freeze. And it didn't actually come too far south. So we resampled 12 of these 18 sites all the way down to Pine Island. This is the Pine Island out near Weeki Wachee, not the Pine Island down by Caloosahatchee, and resampled these 12 sites. And we're able to compare it to the field study of just the previous summer. So we went back to those sites. On the bottom, here's a little timeline. That baseline assessment was July 2022. The freeze happened in December. Immediately the next month after, Calvin and Hannah were out doing the damage assessment. And then importantly, we came back after the next growing season to see whether there was any recovery, resprouting, flowering, fruiting, etc. Out in the field, we looked at leaf damage by stratum, by species, whether it was landward or water side. We looked at severely damaged. We looked at canopy height reduction. That was all during the damage assessment. Then the following November came back and looked at mortality, who actually died and who resprouted. For those synoptical high trees, again, leaf damage, height reduction, vegetation cover, and then also at the recovery, looking at mortality and resprouts. Okay, so a couple of big take homes here. We found that the temperature thresholds for freeze resistance, in other words, how vulnerable were these species to actually getting damaged? Black mangroves had the highest, red next, white next. And this is what that looked like. So here I have T minimum of that freeze event on the x-axis and leaf damage and mortality on the y-axis. And we have these species-specific temperature thresholds, -5.7 and 6.2 for leaf damage and mortality for black mangrove. I'll pop up the red and the white. And we see this general trend of that threshold being lowest for black mangrove, medium for red, and highest for white. We didn't have enough white mangroves in the study to get a threshold for mortality for white mangrove. But this allows us to do species-specific, basically tolerance thresholds that we can map into future mangrove range expansion, structure, and extent. So that's freeze resistance. We also saw that the freeze damage was highest in tall red mangroves. So I'm going to talk through these graphs for just a moment. On the left side is leaf damage, on the right side is mortality, both measures of basically the tree being damaged. We have tall and short black mangroves in gray, tall and short red mangroves in red. And we see that in general, we have the highest damage and highest mortality in red mangroves. What we also saw is that tall and short red mangroves responded very differently. So there's a very strong difference between the damage in tall red mangroves, which was high, and short red mangroves, which was relatively lower. And it was even more stark for mortality, where basically very few of the short red mangroves perished in the event. For black mangroves, although it was slightly higher, it was not statistically significant. So black mangroves are basically taking the same damage, and it's way lower than the red mangroves. We also saw that tall black mangroves had the highest resprout rate. So there is basically no resprouting in the red mangroves. I showed that one picture because it was sort of such a phenomenon, but generally zero resprouting of the mangroves, red mangroves, and much more resprouting in the tall relative to the short black mangroves. So now we're not getting just at what was the damage they took, but what are their modes or methods of recovery and resilience. So we can use this to answer the question how climate will affect mangrove expansion in the future. And to do this, I'm going to show some climate data, I'm going to show some thresholds, and we're going to try to understand what we expect Apalachicola to look like, let's say in 20 or 30 years. So I've got two sites here. On the left is Apalachicola Bay. So this is the red circled area on the map. And on the right is Cedar Key. And all these squiggles and lines are basically their temperature data. They're the annual minimum temperature at these two locations. The green stuff is all observed data plus a range over a range of reanalysis data and location. And then the red and yellow stuff is projected based on different community climate modeling projections out through 2100. So we've got observed data, we've got projected data, and that's what we expect over, through the end of the century. So I take those away, and there's three other important lines there, which are the dashed lines that go across. And those are those temperature minimum, those thresholds from just 2 or 3 slides ago, the 5.9 and the 4 point and the 3 point. So those are the thresholds for black, red, and white mangroves. What that allows us to do is really look out into the future and say, how often are the thresholds for each of these species going to be exceeded? And we do that, we use something called freeze degree days. It's basically the cold that the mangrove feels over a season. And basically, a lot of freeze degree days means either it was above freezing for a very long time, or it was, sorry, below freezing for a very long time, or it could be very low below freezing for a short time. But it's like basically the temperature below freezing times the number of days. More freeze degree days is worse for stuff that doesn't like freezes. So on the left is what it looks like in Apalachicola Bay, on the right in Cedar Key. And we already see stark differences between the two sites, right? One is further north. They have a different climate, different temperature regime. But we can compare on the right sort of the present day, about the present day of what Cedar Key looks like in terms of freeze regime. And it's very similar to what it looks like about 20, 30 years in the future for Apalachicola. We see this shift towards a much lower freeze sort of experience. And so that begs the question, are we going to see this transition as well? And, the transition in Cedar Key from marsh dominated to mangrove dominated was fairly rapid. Happened in, the memory and the lifespan of people who live there, who study there. And so we expect ongoing changes in Apalachicola that sort of mirror that effect. Now, there are a lot of other differences. There's the river there, there's sedimentary differences, but in general, the climate is looking like it's going to be a proxy for what Cedar Key is now. So I want to just do like sort of a conceptual summary of what this whole mangrove freeze resistance and resilience story looks like. We've got the three species, black, red, and white mangrove. We've got tall ones, we've got short ones. This picture is like a cartoon, it's no data. But we have this idea that there's a mix of species, a mix of strata. Then there's a damaging freeze event. And when that freeze event occurs, the black mangroves, we found them to be pretty resistant. They can withstand the lowest of the freeze temperatures in both the short and the tall strata. So they decline, but not so much. For red mangroves, they get significantly damaged, especially for tall trees. The short trees damage, but much less so. So what's left over after a freeze event is sort of this reshuffling of some tall trees, some older trees, and a bunch of short trees for red. For white mangrove, it's the least freeze resistant. We actually don't have much data for the short tree strata for white mangrove, but in general, the least freeze resistant. Then some time of recovery without a freeze event. And of course, these, interfreeze intervals are getting much, much longer as climate warms. But for black mangroves, they have a highest freeze resilience because they resprout like mad. So they resprout and the short trees will be growing. For red mangroves, the tall trees have minimum recovery. They barely resprout at all, but the short trees survive, so they're resistant, and then they can grow back that way. So it's interesting to think about like different modes of resistance, more resistant for black mangroves, and the resilience comes from growth and resprouting. For red mangroves, well, the tall ones are not resistant to the freeze, but the short ones are resilient by being able to survive and grow back. And then for white mangroves remains unknown. There's probably some recovery through resprouting, and we're not really sure what short trees are going to do. So that's basically the two-thirds of the story I wanted to tell. And I said I would leave out the last part there about what's happening with soil carbon, but maybe Laura and Savannah will invite me back next time because this we just got the proofs back for this article showing how soil organic carbon changes as mangroves begin to replace marshes along that temperature gradient. Um, but I'm going to leave you with some take-home messages, basically uh, from those three chapters. The first is that across individuals, populations, and communities, we saw variation in mangrove structure across winter temperature gradient. So that meant how tall they were, how many there were, the biomass, the species composition, and that field data across that chronosequence allowed us to predict future change of Florida's mangrove species on the Gulf Coast. We saw these species and strata-specific responses of how mangroves are going to respond to freeze. So this we call it the freeze-mediated community means, well, of course, we expect there to be some freezes in the future, but less and less frequent, further and further apart, and they allow us to understand this expansion and development in a warming future. And here's my little spoiler about the soil is that we do think that mangrove expansion probably, I said influences here because it's a little bit, there's a little bit of an argument in the literature now about whether it increases or not. But we found some increases in soil carbon storage, but we found regional variation in sedimentary setting. External inputs were also playing really important roles. And so maybe I'll come back and talk to you about that next year. But in the meantime, I want to just acknowledge Hannah Sackles in the field, Ivana Sarah, Alexis Jackson, and Ms. Zhu for help in the field. We got funding from the U.S. Geological Survey, NCBS, and the University of Florida. I was told to move this to the side, not close it. And especially I had to, we had to do a lot of work to get access to all these sites. So thanks to the DEP, Apalachicola, Rookery Bay Estuarine Research Reserves, the Cedar Key and Ding Darling National Wildlife Refuges, and the Cockroach Bay Aquatic Preserve for all that site access. And with that, I'll leave you with this AI image that Calvin made and your poll. This is what mangroves look like under a freezing frost with falling snow. And happy for questions as well.
Thank you so much. Lots of claps. It was a great talk, and we actually already have a few questions in the chat, so I can start with those. If you guys have other questions, feel free to raise your hand or unmute. The first is there any evidence of hybridization among the three different species of mangroves?
From what I know, the answer is no. I have not come across any information about species hybridization. I'm sure that there is sort of, basically adaptation and selection going on where sort of the ones that survive are the most cold tolerant. And, we've seen that with all sorts of environmental gradients. I think you had Carrie Adams last week, so she talked probably a lot about phenotypic and genotypic, selection. I'm certain that that's going on, but if someone else on the call knows about hybridization, I'm happy to hear about it, but not that I've heard of.
I haven't heard of it either. You got to a little bit of this. This was asked before the second chapter, but do you know the mechanisms by which the red mangrove outcompetes the black mangrove at warmer temperatures?
Yeah, that's a great question. I think, basically literature says that there's basically productivity differences. And so as the red mangrove is sort of relieved from the freeze limitation, especially those tall trees getting knocked back by the freezes, they're basically their biomass production, their above ground biomass production rate is really high. And as far as the height, I would think that there's potentially some shading effect, but observationally and even from the data, I said the black mangroves are some of the highest out there as far as canopy height. So I think it's really about basically above ground biomass production. And then when you think about all the other potential mechanisms about just allelopathy and all those other things, I don't think we have a really strong idea of what are the major drivers of that takeover. It's really the release. And the other thing I'll say is like we told the story here, like, oh, red, black, white latitudinally, but we also know there's this zonation sort of from the coast inland. And the way that we set up these transects, they are shore parallel transects, trying to get sort of the mangrove zone. They're fairly wide, but they don't account for things like hypersalinization at perhaps like the high tide marks and things like that. They also don't account for things like we know that temperature is warming, but we also know that sea level is changing. And so those projections are temperature-based projections that would also, you'd imagine, a landward or an inland shift of those communities.
Um, Great, thank you. Under the most ideal conditions, is the maximum mangrove height genetically fixed for each species? And is this gene adaptation slash changing, mutating as a result of climate change?
Oh my gosh, did you introduce me as an environmental engineer? Because so, I love to say I don't know, and when I don't know the answer to a question, but yeah, so I don't know the answer to that question. I'll say something that floors me about canopy height, which is that the mangroves around Cedar Key and the surrounding area, when you look at this threshold, you kind of expect this sort of smooth thing, and then you level out, and you've got this blip, and the blip on top are these mangroves in Cedar Key, and they're like taller than you might expect just from that empirical relationship. They're kind of gangbusters. And I think about the Swanee as a source of nutrients and sediment, but what I haven't found is a really good explanation of what would predict the sort of what do you call like the optimal or like without any other restrictions or limitations, what affects that maximum height. So I'd expect it's like other trees, it is genetically programmed. But now I'm getting out of my expertise.
Well, I think the next question is a little less biology, so maybe good.
Yeah.
Um, Maybe. Please explain a little more about why the main variable of interest was T min and how the results might compare with other temperature variables like average temperature, number of days below freezing, etc. And also, were you surprised by the threshold temperatures in the cover abundance models were all below freezing?
So yeah, great question because I can answer that one. Okay, so yeah, so we actually did an analysis where basically everything I showed you was a graph with something on the y-axis about mangroves and T min on the x. So we did a lot of work to say what were the other things that could be on that x-axis. We looked at average temperature, we looked at um, you know, average yearly minimum temperature or like global minimum temperature over the last 40 years, over the last 30, over the last 20. We did freeze degree days and basically on down the line kind of like you would do to say, like, how do you characterize the stream flow regime, the max, the min, so we basically took that same approach and found that across the board, minimum temperature of the last 40 years was the best predictor. And so that is sort of maybe that's satisfying because it's complete, but it's not satisfying because it doesn't have you know, it doesn't include, for example, duration. If you had a T min of -8 for 1 day versus a T min of -6 for 4 days, you might expect a very different response as the basically plant cells begin to cavitate and everything. Um, but statistically, that's what came out. And I think it's sort of like a bulk measure, but it's really getting at the magnitude rather than the duration. But we do know that extreme freeze events of, more than 2 days really knock back populations. And the second part was, am I surprised they're all negative? Um, no, just from reading the literature, we see that mangroves can withstand brief periods of low temperatures, and that black mangroves, especially in the southeast, have been seen to have threshold responses around that temperature.
Great. And how did you classify tall versus short?
Yeah, this is just, from the mangrove literature. I think it's 1.4 meters and above became tall, and 1.4 below became short. And so they were randomly selected in within the plot.
Cool. Um, there's lots of thanks for the information, and this person is with FWC and manages the Big Bend Wildlife Management Area along the Florida Gulf Coast.
Oh,
Mangroves have
awesome.
expanded into the salt marshes, outcompeting herbaceous vegetation that is critical habitat for imperiled species such as the eastern black rail and the salt marsh, the songbirds. Do you have management recommendations for land managers who are facing these challenges?
Yeah, I mean that's a tough one. And I think Laura and Savannah, we get questions like this all the time, like hey, it's getting warmer, snook are hanging out. Like how do we make sure they're still there? Do we need to change our water management? Do we need to change our vegetation management? I think that, we see climate warming and winter freeze regime changes as like that's occurring. And so it's like that second or third slide I showed that all of the individuals and the species and the landscape are all responding to climate change at the same time. And so I'm not going to advocate, for example, like, so we should go cut down mangroves. I'll just say that very clearly. And I think that is not just for habitat, but also, as coastal residents with marsh views, as their view changes, there will be pressure for that ecosystem service as well. So the view shed changing. And so I think it's probably about educating ourselves and educating others about basically any ecosystem is providing a suite of ecosystem services. And as the foundational species shift, salt marsh to mangrove, you're going to get really big changes. You're going to get changes in uh, basically coastal protection, carbon storage. You're going to get rookeries instead of well, you get different fish habitat. You'll get, in fact, I think I have a slide about this, but it's okay. Carbon short wildlife habitat, fisheries change, wave attenuation, view front, and so I think it's sort of understanding it, trying to understand the timing of it, and um, yeah, that's about all I can say on that one. I'm happy for anyone else to jump in on that who works in this, uh, trying to respond.
We'll move on to the next question, which is, are there any changes in propagule health and disease resistant due to climate change?
Thanks, Diana. Yeah, I don't know the answer to that question. I do know that, just trying to understand the range expansion, we really want to understand whether propagules are available and whether they overcome like the herbivory pressure from crabs and whether basically there's enough being produced. But as far as their disease resistance and climate change, I'm not sure the answer to that.
That's the last question we have in the chat. If other folks have questions, you're welcome to unmute and ask questions directly. But while you're thinking of that and know that there's the opportunity, I'll ask a question. In the southern part of your surveys, they're definitely like it's more urbanized. So were all of your surveys in relatively, big forests, or were they backed up against cities as well, which might impact your results?
Yeah, most of them were in at least, what I'd say would be like couple kilometer wide mangrove zones. So even all the way down to, we were in Rookery Bay, so that was pretty protected. The area around like Cockroach Bay Aquatic Preserve, it's certainly near urban development, but it's a pretty expansive, a pretty wide tract. I'd say probably the place where we had the thinnest sort of mangrove fringe was probably at Deckle Beach, actually, so north of Cedar Key, and that was just because we had to opportunistically looking for mangroves in that location. I'd say all of them were fairly extensive stands. But certainly things like as you go farther south and you're getting near the Caloosahatchee outfall and thinking about, one thing we thought a lot about is just in the north, we've got this sort of sedimentary setting that is, siliclast. It's like stuff coming down from Appalachia through the Apalachicola River. And in the south, we have this carbonate setting and very little sediment. And so there are differences that are, physiographic between the sites. And by sort of collapsing onto that one temperature variable, we definitely ignore that. And I think in the soil carbon chapter, that was where that was the most clearly different. And we're looking at mangrove function in terms of soil carbon storage was most challenged by the fact that south and north were different. As
Yeah,
far as the actual community composition, the structure, I think that all the sites were pretty representative of that region.
cool. Well, I won't ask all the questions I have down here, but I will ask one more. Your observation that tall trees die back more than short trees, is that because it's colder? higher in elevation, or is there something different about those trees?
Yeah, so that I think that remains an open question, and that was just for the red mangroves. But I think, we've done, or others have done some work looking at basically microclimate variation. Do
Yeah.
you basically get some buffering at the ground surface? So that's sort of that's the going theory, is that there's basically a little bit of warmth retained in the soil, and so that those smaller trees are able to, if you had like a temperature profile from the ground, and folks have done this, and they found that basically it's colder as you go up, and we'd expect that.
So in the north where it's cold, can you have really old short trees, or are those short trees always young?
No, they can be old, and there. So one thing I didn't show is that we looked not just at how many and their height and stuff, but we also looked at the number of branches, and we were able to calculate. Well, we had a hard time deciding how to spell it, but shrubbiness, like whether it had a Y or an I, but we had like a shrubbiness index, and a black mangrove of a certain age in Apalachicola compared to one in Cedar Key, much shrubbier. And but they still could, and they're not quite as old necessarily, but they can be old. They don't have to be short and young. They can be short and teenagers. Yeah.
Cool. Okay, well, last call. Anybody else have any questions? I have lots of questions about the ecosystem services, including the carbon. So I hope we can get you to come back next year and hear more about that.
I hope so too, Laura, because I think we got like one step and there's a couple more steps, and like we need to write a proposal or something. So I would love to team up with NCBS and SWES again. That'd be great.
Great. Well, thank you so much for everyone for coming. Thanks, David, for rescheduling with us. Our next talk will be in 2 weeks, and it'll be on prop scars and seagrasses by Annie Hensel. So talk to you guys soon. Thanks so
Thank you very
much.
much. Bye.